Well, thank you, Jose, and thank you, Miguel and Jesus, for the invitation. I've really enjoyed the meeting so far. So I was very fortunate. Dr. Justo Garcia de Jimenez had a beautiful talk this morning providing a lot of background for Huntington's disease. I'll add just a few more points, or at least remind those who maybe weren't there. And then I'd like to talk about a few things that we're doing. One is single-cell longitudinal analysis to measure in the normal population. So in this room, we may have anywhere between 4 and 35 CAG repeats. Is there a button? Which button do you press? But if you have even a single repeat above 35, you're at risk for Huntington's disease. And that CAG expansion gets translated into a homomeric polyglutamine stretch in the full-length protein. And there's still been a lot of work trying to find out what that Huntington protein does. There have been several functions attributed to it, but it's believed that this disease is primarily a gain of function. In other words, there's something about that long polyglutamine expansion that causes Huntington to do something that it wouldn't normally do that causes disease. So you saw a beautiful figure slide before in showing the relationship between the age at which patients develop symptoms and the length of the CAG repeat expansion. This slide came from Jim Gazzella. And the important point was made earlier that although there is a strong relationship, in general, the longer the CAG expansion, the earlier you develop symptoms, there's quite a bit of variation, especially around the CAG repeat that corresponds to the disease threshold. But I did want to make one other point. This blue line here and the red line here represents the number of alleles, Huntington alleles in the general population. And so the point is that most people with Huntington's disease fall into this large bell curve and have CAG repeats in the sort of 40 to 50 range. And it's rare to have these very long expansions. But there are people with intermediate expansions that are technically normal, but there's a view now that this may be a pre-mutation range and that it is possible for people to spontaneously have that expand and develop Huntington's disease. So one of the, as Jose mentioned, one of the reasons I got interested in Huntington's disease was this puzzle about when we look through the microscope, and this applies for whether you're studying Alzheimer's or Parkinson's or Huntington's, ALS, any of the neurodegenerative diseases, how do we understand the biological significance of the changes that we see? Because when we're doing pathology, we're limited. We're taking a snapshot in time, and then we're trying to infer what is going on biologically from that static snapshot. And when we began, there had been a publication suggesting that these neuronal internuclear inclusions which get formed from Huntington was the cause of Huntington's disease. But we and others, including Harry Orr and Huda Zogby, had done a number of experiments where we manipulated various aspects of the disease and could dissociate the formation of these structures from neurodegeneration. So we began to ask, what really is the role they play? And we realized as we thought about that, we couldn't even answer a very simple question with these conventional approaches. For example, I couldn't even tell you whether a cell that formed one of these structures, what happened to it. Did it live longer? Did it live shorter? I had no way to know this. And as I thought about this even more, I think it really began to sink in that the way I thought about disease had up until that point been shaped significantly by pathologists who had carefully described oftentimes changes in tissue as disease unfolds. And it was my maybe false assumption that those represented steps in a linear process of pathogenesis. But I think what we've learned in the intervening years is that pathology doesn't equal pathogenesis. It's oftentimes much more complex. And some of the changes that we might have initially thought were actually pathogenic turn out to be incidental when we have ways to really understand their role. And in other cases, changes that we thought were pathogenic could even be coping responses by the tissue or cells to try to deal with the problem. And so we really struggled with this. If we had no way of really understanding with conventional approaches using snapshots in time what role these different steps were, what could we do? And so a postdoc in my lab at that time, Dr. Montserrat Arasati, and I worked together to try to develop a new imaging system that would allow us to watch this whole process as it unfolded over time and be able to develop a way to measure when these intermediate steps appeared and what their relationship was to a fate that we cared about. So in this next step, I'll just tell you a little bit about that system and how we've applied it. So this is a picture of Monsi back when she was in my lab. She was trained by Dr. Jesus Avila here, and she's currently running her own laboratory at the University of Navarra. She's a terrific person. So she helped us generate the very first system we've made, and then we've continued to develop that technology, and members of my lab generated a second generation system. We just finished a third generation system in December. The first was developed about 10 years ago, and we're now building a fourth. So the third system begins with a robotic incubator. So there's a robot in the incubator that will pick out the plate that we want to image. There's a barcode scanner there that will check and make sure it's the plate we want. Then we've integrated a robotic arm into the system that will transfer the plate to and from the incubator. And then the microscope itself we've built so that it's fully automated. We have motors on the focus knob, we have motors on the stage, and it automatically focuses by itself. And then we've written programs to coordinate all those movements so that the instrument can do automated imaging 24 hours a day, 7 days a week. But the special thing, so that, what I've described so far as a high throughput imaging machine, the special thing is that we wrote programs that allows the microscope to know where that plate is in three-dimensional space. So when the plate gets taken off, it can be returned the next day, and you can go back and look at exactly the same cell. So if you look at this bottom movie here, what we've done is just taken one picture per day in a high throughput manner and then concatenated them into a movie. And so you can get a sense of just how finely aligned the images are. It's really possible to go back and follow the lifetime of individual cells. We've followed cells as long as 6 months. You can follow them as long as you want with this system. And this third generation system actually has its own Twitter feed, so it'll tweet you when your experiments are done. So the last couple of years, we've really worked hard on developing programs that will analyze all those images. So a typical instrument generates about a half a terabyte of data a day. And so one of the things it does first is stitch the images together into a montage. And then the computers will go through and find the individual neurons and give them little social security numbers, each of them, and then extract fluorescence data from those cells and track it to a spreadsheet. It'll go through each of the days in the same way, and if a cell's died, it'll mark the time of death. And then I just want to emphasize that although all these images just showed a sort of gray-white spot for a cell, in fact, we can put different fluorescent proteins into those same cells at the same time and measure independent measures within the same cell. So we can get measures of morphology and viability. If we've tagged the protein that we're studying, we can measure levels of aggregation. We can put in reporters to look at different functions. And so with this system, it's possible to measure all of those dynamically over time and then relate them on a single cell basis to the fate that that cell then experiences. About 4 months ago, Google came to us. They were very interested in the platform, and they've been very helpful in helping us manage the data and developing. They've assigned us software engineers to develop even new algorithms. And one of the ones I'm most excited about is the integration of machine learning into this approach. So as a human, we know these images are very rich. They have a lot of data. But as a human, we're pretty limited in terms of the sorts of analyses that we do. We may count cells or count synapses or mitochondria or things like that. But I'm sure there's more information in those images than we are really getting out of them with our simple forms of analysis. And with machine learning, what we can do is we can begin to allow the machines to look at the images themselves, extracting many more features than we could as a human, thousands, and then begin to relate them to each other and to fate, to be able to tell us as experimenters what is the most interesting thing to look at. In addition, one of the beauties too of this is that we can begin now to integrate disparate data streams. So although this is really focused on the imaging, for some of our cells, for example, we've done whole genome sequencing on them. I've actually seen the patients and I've done clinical phenotyping on them, so I can include those data as well. So in a nutshell, we like to call this clinical trials in a dish because we use basically the same math to analyze these data as we would for a clinical trial, but we're using individual neurons instead of patients. And the key capability is that we're able to follow single cells, be able to measure changes in each cell during their lifetime, and then find out what prognostic value they have on a single cell basis. And with that approach, be able to unravel some of these difficult questions in neurodegeneration. It's about 100 to 1,000-fold more sensitive at detecting changes than conventional snapshot approaches. And that's made it possible to begin to develop it, as you saw, into a high-throughput screening device where we can do genetic screens, for example, in mammalian cells as well as small molecule screens. Now, in a typical experiment, what we'll do is we'll observe fate with the experiment, and we may have an idea of a variable that's important for fate in those cells, and we can test its relationship. The math is pretty simple. It's shown in the bottom here. But we recognize that with just a single variable, we're unlikely to explain much fate, and so you may get quite a bit of scatter. But the beauty, I think, of the system is the math is expandable. So as we begin to identify more and more variables that contribute to the fate of that cell, we can start to build a model, a statistical model, to explain the fate. And our fantasy is that someday we'll be able to write an equation that can explain the fate of each cell just with a handful of variables. And if we can get something close to that, I think we can answer two major questions. What are the major determinants of fate in, for example, cells that have mutant Huntington in them? And then how might we cleverly target that in a rational way to be able to achieve the maximal therapeutic effect? So we have this nice tool. I'd like to just now say a little bit about how we've applied it to Huntington's disease. I'll fairly briefly go over some of the discoveries that we've made in the past and then end with some of the more recent work, including some unpublished studies. So for many years, the main model we used was a primary murine model. We would grow neurons from the striatum, the most vulnerable region of the brain in Huntington's disease, for about a week in a dish, and then we would introduce into those cells versions of Huntington, a fragment or a full-length form, tagged with a fluorescent protein. And then other fluorescent proteins would be introduced to measure other features, for example, whether the cell's alive, whether the neurites are there. And then we would subject those cells to that automated imaging and longitudinal analysis. And I won't take the time today, but we've been using this model now for many years, and we found that it recapitulates 18 features, at least so far, of Huntington's disease at a molecular level. And there, at least 8 times we've made discoveries first with the model that we've then tested in vivo. So although in vitro models certainly have their limitations, it does seem like this recapitulates enough of the disease to be a useful tool to try to understand some of the processes. So this would be a typical set of images that we would collect in an experiment. Normally, for an experiment, we would collect data on maybe thousands of neurons. We've collected data on millions, probably billions of neurons over the years. But I'm just going to show you examples, pictures from three, just to make a few points here. So here are three striatal neurons that have Huntington in them with a green fluorescent protein tag. This is a mutant form of Huntington, together with red fluorescent protein. We use the red fluorescent protein just to mark that the cell's alive and to visualize its neurites. And if you watch over time, you can begin to see things like inclusion body formation occur, as well as cell death, which is indicated here by the disappearance of the red fluorescence. Now, if we'd done this experiment the old way, which is to just take basically snapshots, it would be easy to come to some very different conclusions. So we start out here, we introduce our proteins, and we wait several days, we fix the cells, and we immunostain them. We see that one cell's died and that two other cells have now inclusion bodies. So inclusion bodies increased, cell death increased. It would be really tempting to come to the conclusion that maybe there's a causal relationship. But if we look carefully at the whole set of experiments now, if we really watch things from beginning to end, what we discover instead is that cell that died first actually never formed an inclusion body. And the cell that lived the longest, this top cell here, actually was the first to form an inclusion body. So one of the ways we used this early on was to finally address that question that nagged us for so long. What is the role or the relationship between the formation of these inclusion bodies and the fate of the cell? And what we found was that when inclusion bodies form, cells survive actually better. So it dramatically reduces the chance that they'll die in the next image period. In fact, it only almost reduces it to baseline levels. Now, one limitation of this approach, well, several limitations. One is that these are, mouse cells. They're not human. Maybe there's a difference between mouse and human. We're just using a fragment of Huntington. Maybe it's important that there's a full-length form. We've tagged it with a fluorescent protein. So one of the things we've been really busy doing, especially in the last 4 years, is to try to develop complementary models using induced pluripotent stem cells. You heard a little bit about this from Ken Kosick earlier and from Larry Goldstein. But let me just say a few things if you weren't here. So with this technology, and I should say that my colleague on the floor above me, Shinya Yamanaka at Gladstone, won the Nobel Prize for this last October. What he was able to show is that you can collect a skin sample, so just the live cells beneath the surface of the skin, fibroblasts, and now actually we can do this with white blood cells from a blood sample. You can grow those primary cells in a dish and then reprogram them using a set of genes so that they become basically induced pluripotent stem cells. And then once we form those cells can be passaged, propagated, they can be frozen down and then used at some later date, and we can take those out, grow them, differentiate them to become different types of cells, including the same types of neurons that are vulnerable in Huntington's disease. The idea with development was that stem cells sort of sit at the top of the hill and they roll down to become differentiated types of cells. But what Shinya showed was that you could introduce these genes and basically revert those cells to the stem cell state. And then once they're there, we can use recipes of factors to start to make them into the other cell types that we really want to study for disease. So Dr. Jimenez made the important point earlier on that although we think of Huntington's disease as a neurodegenerative disease, it does have some peripheral manifestations. So one of the things that we've done also is to make these cells from Huntington's patients into different types of tissue. This is an example of cells that were turned into cardiomyocytes that were just skin cells a couple of weeks earlier. And you may be able to appreciate there's some blood cells pumping through a vessel here in culture. And we're starting more and more to make organoids begin to build higher three-dimensional structures. Here's a chambered structure from cardiomyocytes. And so this approach has been pretty successful. We have now made human neuron models of Parkinson's disease based on synuclein triplication and LRRK2 mutations. We've also made human models of ALS, both making those iPS cells into motor neurons as well as astrocytes. And that allows us, when we put them together, also to look for cell non-autonomous interactions that might be important for disease. But the first model we made was a Huntington's disease model. And so we collected cells from patients with Huntington's disease and made them into iPS and then have studied them. And interestingly, the iPS cells differentiated into forebrain neurons or into medium spiny-like neurons have a higher risk of death. So this is the way we present some of our robotic microscopy data. It's basically the probability that a cell in that cohort will die in the next interval. So the higher the risk, the higher the rate of death. So in cells that have 180 CAG repeat expansion, you can see there's a high rate of death, those with 60 a lower rate, and then we get back down to baseline with patients that have a 33 expansion. And the point I wanted to make here that loops back to the talk as I had before this departure was that we looked really carefully, and we did not see any inclusions, or very few, in these cells. So the point was that here we're now using a human neuron from a Huntington's disease patient where we're expressing Huntington under its endogenous promoter in its full-length form at endogenous levels with no fluorescent protein tag, and yet we can clearly see inclusion bodies aren't required for this polyglutamine-dependent death. So we don't think that basic observation is somehow an artifact of the system we're studying. So if inclusion bodies aren't the pathogenic species, what is? And one of the things we were struck by was the possibility that maybe it was the forms of Huntington that were elsewhere in the cell that we called, for lack of a better word, diffuse mutant Huntington. So to try to get at this, what we did was we measured the fluorescence of the tag attached to Huntington, which we had shown was closely related to the expression level of Huntington, and asked whether it was a good predictor of fate in these cells. And we found that it was. For cells that were expressing mutant Huntington, we saw a strong and significant relationship between the level of diffuse mutant Huntington and whether and when that cell would die. There was no relationship with wild-type Huntington. So we were wondering if there was something about the protein itself that maybe changing conformation, even in that diffuse form, that could be important for predicting whether the cell died or not. So one of the next things we did was try to see if we could raise conformation-specific antibodies that might recognize a particular form of Huntington in that diffuse pool that was especially predictive. And what we found was that there were a series of monoclonal antibodies we discovered. We compared a number of them, but there was one that really emerged as highly predictive of whether and when these cells would die. And we went on to solve the crystal structure of that antibody and then to solve a low-resolution structure of it in complex with Huntington. And what we discovered was that it was recognizing a compact structure of the polyglutamine stretch in monomeric Huntington, suggesting that Huntington may undergo a conformational change as that expansion grows that confers to it a structure that may be especially toxic to cells. So with this, we were able, and with that antibody, able to actually do a pretty good job predicting neurodegeneration for any length of the polyglutamine stretch. So from the standpoint of Huntington, did a pretty good job. But still, we only predicted about 50% of when and whether the cell would die. So one of the things we started to wonder was whether there was something other than the protein that was important for determining whether cells were susceptible. And that's when we started to really focus on the environment. In other words, features of the cell itself that might affect how it handles mutant Huntington. And one of the first clues was, back in some of our original images, we noticed that at about 5 to 10% of the time, once an inclusion body would form, cells could spontaneously clear it. So this cell remains alive and continues to remain alive. And so that was pretty remarkable because these are really big structures. And as far as we know, the only pathway that we're aware of that's capable of clearing such a big structure would be something like autophagy, which we heard about earlier. And autophagy is a protein clearance pathway that cells normally used, but maybe it's more engaged when you have misfolded proteins on board. And so we got really interested in the idea that maybe inclusion body formation is just part of a broader coping response that neurons unfold in response to misfolded proteins. And so with that, we started to really focus in on the clearance pathways that neurons manifest. So one of them is the ubiquitin proteasome pathway. And there'd been some controversy in the field about whether this pathway was inhibited by mutant Huntington or whether proteasomes were capable of clearing mutant Huntington. And I must say, if we had to study this using just cross-sectional approaches, it would have been very difficult. But with single-cell approaches, what we found was that the proteasome does seem to, that its function appears to be impaired in a matter of speaking before inclusion bodies form. But then when inclusion bodies form, at least that functional measure improves. But it's very time-locked to the process of inclusion body formation, which itself isn't synchronized. So it's something that would have been lost in the noise, but with single-cell analysis, we were able to see that relationship. But as I mentioned, autophagy looked like it might also be involved, and so one of the things that we've done, and I'll finish my talk later on talking more about this, is to really begin to focus on that process and to see whether that might be a therapeutic avenue that we could exploit. So we got very interested since I just shown you that levels of mutant Huntington seem to be really important for determining whether and when neurons die. And there seemed to be evidence from studying the neurons themselves that they responded to mutant Huntington by generating protein clearance, activating protein clearance pathways. We got really interested in understanding what controls the metabolism of Huntington itself. So a postdoc in my lab, Andrei Svetkov, began to look at this. And the challenge was that at the time, it's very difficult. The standard approach to measuring protein metabolism is something called the metabolic pulse chase analysis. So with this analysis, you isotopically label your cells, and then you, at some time point, lyse those cells, solubilize the protein, and then measure the radioisotope that still remains. And you can use the disappearance of the radioisotope from your protein as a measure of how long-lived that protein is. Now, the problem for us is that in studying aggregation-prone proteins, particularly toxic ones, there are two confounds that get introduced. One is the aggregation process. So to the extent a protein aggregates and is difficult to solubilize, that signal will be lost, and so we will underestimate the half-life of the protein. Similarly, if the protein's toxic and the cell dies between the time the cell's labeled and the time we make our measurement, it also will lead to an underestimate. So for those reasons, it's made it actually quite difficult to get really good measurements of half-life of Huntington. So the approach we took was to try to see if we could develop an imaging way to do this. So we created a method we call optical pulse labeling. It turns out that there are a number of fluorescent proteins now that have a really interesting biophysical property that makes them photoswitchable. So in this case, it's a protein from a coral species called Dendra-2, and it starts out as a green protein, green fluorescent protein, but if you pulse blue light on that green protein, it will irreversibly change to red. And so we can use that as basically an optical label that we can then watch as it gets cleared from the cell. So for example, we replaced the GFP tag on Huntington with Dendra-2, and we introduced it into neurons. And so before photoswitching, all you see is green signal and no red. But if you photoswitch those cells, now a pool of that green protein becomes red. And as long as we don't do any more photoswitching, that is a fixed pool, and we can watch as it gets cleared from the cell and measure that as a surrogate for its metabolism. So we can use the microscope to go back to that same cell and see as the red fluorescence disappears, and we can compare the clearance rates of Huntington when it has a polyglutamine stretch in the normal range versus that in the disease-causing range. And the first surprise was that the half-life of mutant Huntington is actually shorter than the half-life of wild-type Huntington. We'd expected, since Huntington accumulates in brain over time, that it might be longer. It's nearly about a fifth of what the wild-type protein was. So we started to get a little worried that maybe this was, that our method wasn't giving us the right answers. And despite all the caveats I told you before about metabolic pulse chase, we did find a situation with using mice with full-length Huntington expressed at endogenous levels with a long expansion. If we went early enough, so within the first couple weeks, we could do metabolic pulse chase on those cells. And basically, we saw a very similar result. In mutant Huntington was a significantly shorter half-life than wild-type Huntington. So it's exciting in the sense that what it's telling us is that neurons are able to make a distinction between polypeptides that are otherwise identical except for the polyglutamine expansion, and they're able to target the disease-causing version for more rapid degradation. So what pathways do neurons use to clear Huntington? To try to address this, we went back to our Dendro2 system, growing striatal neurons, and then added selective inhibitors of the proteasome or of autophagy, and then tested what effect they had on the half-life of the protein. And interestingly, what we found was that the half-life of mutant Huntington was much more sensitive to autophagy inhibition when it was mutant Huntington, whereas wild-type Huntington seemed to be cleared more by the proteasome. Dr. Javier Ruiz this morning's talk mentioned NRF2, and I noticed there are like 3 posters out there on NRF2. So hopefully this will be familiar to a lot of people in the audience. This is a transcription factor that was originally identified for its role in managing reactive oxygen species. But I think more recently, people have recognized that it plays very important roles in mitochondrial biogenesis and bioenergetics, as well as regulating even autophagy. So NRF2 regulates P62, a gene that's involved in autophagy. And so we've been interested in its role in protein homeostasis. So what we did was to introduce NRF2 into neurons, and then we measured the half-life of Huntington in the presence or absence of NRF2. And what we found was that when NRF2 was present, the lifetimes of the proportion of cells with shorter Huntington lifetimes was greater. So in other words, it sort of left shifted the half-life of Huntington. In other words, it's accelerating the clearance of mutant Huntington. And when we did this, we also saw a dramatic improvement in survival. So the cumulative hazard with mutant Huntington is normally here. It drops essentially to baseline levels when NRF2 is introduced. So it's highly neuroprotective, at the same time that it's accelerating the clearance of mutant Huntington, lowering its levels. So we think that protein homeostasis is that the cell's response to mutant Huntington is a really important component for determining whether it's going to die. I did want to say one other thing, though, and this was sort of a serendipitous observation. My training in biochemistry was that a protein half-life is a single number that gets assigned to a protein, and it's based on these population measures. What we found, though, when we studied single cells was that there was quite a bit of variability among individual cells. So the same polypeptide, but different half-lives. And so, again, sort of worried that it might be something with the method, maybe some sort of relationship to overexpression, but that didn't seem to be the case. We could find examples of cells where we overexpressed, in this case Dendro2, 100-fold more and get essentially the same half-lives. And at the same time, we could find examples of cells where the levels actually started out higher, but they had a shorter half-life, and others where they were lower and had a longer half-life. So what it suggests is that what we're really measuring is the endogenous protein homeostasis capacity for that cell, and that there may be some variation in proteostasis among individual neurons. So to get at that a little bit more, we actually measured half-life for a model substrate, Dendro2, in a population of neurons, and we're shocked to find that it could vary up to 4-fold. So even though each measurement was quite tight, the actual half-life that we measured for individual neurons could vary substantially. We weren't expecting that. And so to get back to this issue of cell specificity, why do some cells die in disease and others don't? we're intrigued with the possibility that maybe there could be some cell-specific variation in the ability of, for example, striatal neurons or cortical neurons to clear proteins that misfold, and maybe that could be related to how susceptible they are to different misfolded proteins. So we grew striatal neurons, so the neurons from the most vulnerable region of the brain, as well as cortical neurons, which also are susceptible, but somewhat less so. And then we introduced into them the very same versions of Huntington and measured the half-life. And surprisingly, we found that the half-life of mutant Huntington in the more vulnerable neurons was longer, significantly longer, than it was in cortical neurons. And we followed those cells prospectively then, and then determined their fate. And what we found was that to a great extent, we could predict their fate based on the half-life of the protein in that cell, no matter whether the cell came from the striatum or the cortex. And we've done sophisticated Bayesian hierarchical analysis to really kind of quantify these relationships. And we've compared Huntington, the sort of significance or predictive value of Huntington dose, that is the amount that the cell starts with, compared with its clearance capacity. And what we find is that clearance capacity for a cell is about 3 times more important than the dose in determining whether and when that cell will die. So, you know, for many of the initial years, we were very focused on the Huntington protein, but what these data are telling us is that the cell's ability to handle that protein is also a very important determinant of whether that cell will succumb to mutant Huntington. So to summarize sort of a model, we think that environment or mutations make certain proteins prone to misfold. These in turn can adopt structures, it could be oligomers or even toxic monomers, that can place stress on protein refolding and clearance pathways. And that reduces the viability of those cells. In that context, inclusion body formation may be a way to remove those toxic conformers into a structure that's more inert and decompress the stress on the refolding and clearance pathways. And it also suggests that this may be an opportunity to intervene therapeutically. Perhaps there are ways we could stimulate these refolding and clearance pathways. You heard from Dr. Lucas about ATF5, and one of its targets is HSP27, a small chaperone, and so maybe these are promising pathways. Now, I told you that there seem to be some differences between striatal and cortical neurons. We think the basic structure of this model is correct for both cell types, but there are also cell-specific factors that seem to influence the kinetic rates of these different steps. I didn't have time to show you today, but we found that cortical neurons are much more readily form inclusion bodies with the same dose of Huntington than striatal neurons. So clearly there are some cell-specific factors that affect the rates of some of these processes. And we have some independent evidence that protein clearance may be very important. We've done some family-based whole genome sequencing for Huntington's disease families. We've done 51 genomes. We've identified several modifiers, and one of them appears to be a modifier that's involved in proteasome function. So using that basic blueprint for developing therapy, we really started to get interested in clearance pathways, and especially autophagy, which you heard about this morning from Dr. Lucas and some others. So just to remind you, there are 2 major clearance pathways in cells. There's the ubiquitin proteasome system and autophagy. And the distinctive feature of autophagy is that this is a clearance pathway where you form membranes de novo that engulf cargo and target that cargo for degradation by fusion with lysosomes. And one of the reasons that it's attracted the attention of people in the field of neurodegeneration is that it's uniquely maybe able to handle protein aggregates. So once a protein aggregates, it can't fit through the ubiquitin proteasome. So to be processed by the UPS, the protein needs to be unfolded so it can be threaded through the pore of the proteasome. But autophagosomes are able to engulf aggregated protein and clear them. I think there are a couple other reasons to be interested in this process. One is that mitochondria, that's the main mechanism by which they're turned over. It's called mitophagy. And mitochondria, as you know, show up in Parkinson's and Alzheimer's and Huntington's disease as potentially common threads between these disorders. So perhaps through using up autophagy capacity with protein aggregation, we're somehow interfering with mitochondrial function. So we, a few years ago, described some small molecules we discovered in a candidate screen that stimulate autophagy in neurons. These work through an mTOR-independent pathway. A number of them are FDA-approved drugs. They can be taken orally, and they cross the blood-brain barrier. And so we've used those and used computational models to model the active structure of these small molecules in silico, and then use those in silico to do a 1 million compound screen. We've identified many other autophagy inducers that way, some that are more potent and safer than the ones we found initially. But one of the hurdles, or one of the difficulties in trying to do small molecule development for autophagy is that the assays we have for autophagy are pretty poor. So it's either electron microscopy, which of course is very slow, or Western blot assays, which tend to be variable, insensitive, and again, we're stuck sort of taking a snapshot of what is really a flux. So it's really a dynamic process. So one of the things we did recently was to develop a high throughput assay for autophagy. And it uses the same principle that I mentioned to you before. But this time we attached Dendra2, that photoswitchable protein, to LC3, which is a cargo for autophagy. So with this, what we can do is we can put cells, this reporter in, do our photo switching, and then watch the clearance of this. It's a surrogate for autophagy flux. And so we can get a measure of how fast this reporter is, the half-life normally, and then we can add a gene like Beclin, which is known to stimulate autophagy, and we can show that with this reporter system we can accelerate autophagy, basically cutting the half-life of this in half. And our small molecules basically do sort of similar things. So one of the things that we've done is to test these now in our human neuron models of Huntington's disease. Here we're using our iPS made into forebrain neurons and can show that when we add our drug, we can significantly improve survival, reduce the hazard curve here, and it doesn't seem to have any effect on the wild-type cells. Now, our lab has sort of branched out from Huntington's disease to develop ALS models and Parkinson's disease models, and so one of the things we've been interested in is this protein dyshomeostasis a common thread? And one of the things we've noticed as we've studied our LRRK2 models, synuclein, TDP43, is that in none of these is inclusion body formation required for the death that we see. And in all of them, those diffuse levels of the protein seem to be important. So one of the things we wanted to do with these autophagy inducers was to also test whether they might be beneficial in other forms of neurodegenerative disease. So with our human ALS model, these are human motor neurons that have from patient with a TDP43 mutation, we again see that autophagy inducers significantly improve survival but have no effect on the wild-type cells. And it's not just survival. We've looked for intermediate phenotypes like neurites. So normally, over time, neurites from these cells grow and they become very dense. But in the TDP43 model, what we find is they start out somewhat less, but then they degenerate pretty rapidly, and it's not very subtle. So we've used that in an automated assay to be able to measure and watch as neurites grow normally, as they degenerate in our TDP43 model, and then as we add our autophagy inducer, how we're able to restore, at least mitigate some of that loss in neurites, which is encouraging. And we've even made attached our photoswitchable protein to TDP43 itself to see if adding these autophagy inducers actually accelerate the clearance of this mutant protein. And with this sort of approach, we get these single-cell measures of TDP43 clearance. We can replot those as basically a distribution, which each of these hash marks is an individual cell. And what we found is that normally it has sort of this distribution, but when we add our autophagy inducers, we can shift that distribution left. So we can shorten the half-life of TDP43 by half. So we're really interested in the idea that there are actually some common threads to neurodegenerative diseases. I'm sure that they have initiating factors that are largely distinct, But maybe protein dyshomeostasis is something that's involved in a number of these diseases and may give us an opportunity to intervene in a way that could be beneficial to more than one disease. And maybe one of those strategies is to target autophagy. So just to summarize, I think neurodegenerative disease is a dynamic, complex interplay of pathogenic insults and adaptive and maladaptive coping responses. So a key future therapeutic challenge is going to be to identify pathways that explain fate and that can be manipulated safely and effectively. And it's, our data would suggest that proteinopathies like Huntington's disease may in part result from stress on the protein homeostasis system and therefore benefit from stimulation of protein clearance. We're really interested in a number of future directions. I think one is to begin to think less about these diseases as individual molecules, but more as a system, and really begin to understand the dynamic interplay between these pathways and molecules and systems. So to think about it more as a whole, and hopefully that'll be more productive. Also, can we change the way we screen? Can we maybe use this more systems approach as a definition of disease so that we can find molecules that don't just fix one thing, but really fix the disease in a more global way? And I think it's too early to say, but I'm really interested in the idea that these induced pluripotent stem cells maybe will lead us to drugs that are more effective in clinical trials. And I've been really fortunate to have a terrific group of people. Mansi was just one of the great postdocs in the lab. We've had a number of people who've made huge contributions, many of them I mentioned during the talk, as well as the consortium of investigators in the United States and in Europe who have been terrific, especially for the IPS work. So thank you very much.
So thank you, Steve, for this impressive talk, and I'm sure there will be questions in the audience.
So let me see if I have understood well your model, your concept of the difference clearance potency of cortical neurons versus striatal neurons. I wonder if you have studied that in a more general way, because I'm wondering if indeed what is happening is that somehow the interference of Huntington with the proteasome is, let's say, stronger in the striatal neurons than in the cortical neurons. So it's not a question of clearance ability, but sensitivity to Huntington, I wonder.
Yeah, we haven't looked specifically at the proteasome in those two different cell types to see if we see a difference in the way it behaves. But we have been able to measure, essentially, with this single cell analysis, what you can measure is the probability that a cell will die in a certain period of time after you observe it. And so what we can measure is, with the fluorescence of the tag attached to Huntington, we can actually measure the sensitivity of the cell to a particular level of Huntington that's in that cell. And what I can say is that when we compare striatal neurons to cortical neurons, they don't seem to be any different in the sensitivity of the molecule itself. So there seems to be some other, for a given dose, so there seems to be some other mechanism by which the downstream effects. We're very interested, though, in what is the machinery that's important for clearance or for refolding or for other things between these two cell types. Because like I said, one of the most dramatic things is that if you measure the probability that a cortical neuron will form an inclusion compared with a striatal neuron for the same dose of Huntington, it's 3 times more likely to form an inclusion. So there's something about cortical neurons, at least at that level, and their ability to recognize misfolded Huntington and trigger an inclusion body that seems to be different than striatal neurons.
It's an impressive talk, really. Let me ask you, Well, I have many questions, but I will reduce some of them. When you saw the data on the past experiments on the Huntington expanded or non-expanded, that was the GFP or the Denver construct, or was just the, I mean, the normal one, the normal protein?
You mean for the IPS experiment?
Yeah, when you saw the slide with the half-life to check that the half-life of the protein by pulse chase, is that
yes,
the
that was untagged, full length.
So, you know, tagged and untagged may be quite different.
Yes, so that's why, partly why we did that experiment. So we did it, you know, we were trying to compare. Actually, it turned out that the half-life was surprisingly similar, really surprisingly similar. But I think probably more important was that whether the protein's tagged or not, the mutant Huntington seems to be cleared more rapidly than wild type.
Yeah, what I'm saying is that the GFP or the Denver are a long-life protein, so you are fusing long life with long life, you get long life. That is, so the interaction is difficult to understand.
Right, but you understand that the metabolic pulse chase was untagged, yeah.
Yeah, that's the important thing. So another question is, how do you control whether you have, a loss of fluorescence of the protein instead of being degraded?
I didn't quite hear the last part. Control for the loss of the protein?
The loss of fluorescence, because the protein can get in a compartment where the fluorescent protein, the fluorophore, don't work.
Oh, I see, yeah. Back when we did our first paper, we did a very thorough correlation between the loss of GFP and other things, like ethidium homodimer. If we add it to the media, does it stain the nucleus? So normally that dye is kept out of the cell because of the plasma membrane. But if plasma membrane integrity is lost, then the nucleus can stain. We also looked at annexin 5 staining, which is another marker for cell death that marks whether the plasma membrane has become symmetric. And what we found was that there wasn't a single example where the fluorescence was lost in the cell for our viability marker where the cell hadn't died. So we couldn't find any examples where just the fluorescence, where the cell remained alive but the fluorescence was turned off. So it seemed like it was a very tightly correlated with the loss of plasma membrane integrity and cell death.
My question was not on that. Because I have no doubt with living or not living cells, but with the protein itself, with the probe. If the probe lost fluorescence because it's an acidic media around the protein, you will lose fluorescence, but the protein is not degraded.
Well, sure. So we're very careful with the versions of the fluorescent proteins we choose to be sure we understand the environmental sensitivity. And so we try to choose proteins that are relatively pH insensitive, if not completely pH insensitive.
And my last question, because I was so fast that I didn't realize, what was your conclusion respect to TDP-43? It was the aggregated form, the one that goes to autophagy, or it was the diffuse one that goes to autophagy pathway, or it was to the opposite to the proteasome? I didn't catch what were your conclusions.
Yeah, so what I showed with the TDP-43 data was just that when we add, for the Dender2, yeah. So what we're measuring there is just the diffuse form of TDP-43, so not the inclusions. But I can tell you that when we add autophagy inducers, we do see that both the diffuse levels of the protein are lower, as well as the cells are less likely to form inclusions, and oftentimes inclusions that have already formed are more likely to disappear. So we suspect that by clearing the diffuse form of the protein, you are providing less misfolded TDP-43 for the formation of inclusions.
Thank you very much.
Yeah, you're welcome.
So, any other question? I have a quick one, very short, because we are running behind schedule. I found very promising the screening of compounds that increase autophagy rate and the potential in your screening with the different cell models of the ALS and so on. But are you running in parallel an estimate of by itself in wild-type cells how toxic it may be, because it's demanding energy from the cell, because those that are less toxic in combination with the positive effect will be probably best candidates for the clinic in terms of preclinical in culture studies. Have you exploring that?
Yeah, so what we've found so far is the toxicity of the compounds that we have seems to be more related to off-target effects than on-target effects. And we've now got autophagy inducers that work in the sort of 5 to 10 nanomolar range that are capable of stimulating autophagy. And there, we really don't see any toxicity from the compounds until you get to very high concentrations. But yeah, it's an important point. And I think the way we approach screening, it's really, you know, particularly we do all our screening in primary neurons, either mirroring neurons or iPS cells, so it's expensive. So we do tend to first screen against one phenotype, like autophagy flux, and then we will come back and look at effects on toxicity separately. Yeah.
Thank you.